Welcome to this, our fourth in the series of Reverse Rett Emerging Gene Therapies Bitesize sessions. I'm Catherine Sim. I'm the Operations Manager at Reverse Rett and parent to Georgina, who has Rett syndrome. She's 28 years old. We're living in intense times with gene therapies for Rett syndrome now in clinical trials for both adults and children. At Reverse Rett, we have planned these monthly sessions for you, the families and carers of people with Rett syndrome, so that we can share information about what is happening and what we expect to happen in the coming months in plain English at a level that is understandable. We try to keep these sessions bite-sized, which means we start on time and finish on time, answering as many questions as possible. We will be recording today's session. We do ask that you do not make or disseminate any of your own recordings of the webinar for legal reasons. Please keep yourself on mute throughout the session so that the sound quality is as good as possible. We now ask that you flip on your cameras for just a few moments for security purposes. We know that some people are often listening in on the fly. Lastly, if you have any questions you'd like to ask which you weren't able to send in advance of the session, please put those in the chat and we will consolidate questions and ask them at the end of the session. With that said, I'm now going to hand over to my co-host, Rachel Stevenson.
Thanks, Catherine. Hello, everyone. What a start to the year it's been for everyone who's affected by Rett syndrome in the UK, with not just one, but two gene therapies announcing that the UK regulator, the MHRA, has given the green light for UK clinical trials of the two gene therapy products in children with Rett syndrome to go ahead. Our guests today are from one of those gene therapy companies, Taysha Gene Therapies, who last year made Rett syndrome history by starting the first in-human study of a gene therapy for Rett syndrome in adults with Rett syndrome in Canada. That trial is ongoing, and this month the company also announced the dosing of the first pediatric patient in the USA. Our guests today are Taysha's Chief Medical Officer, Bennett Meru, and Senior Director of Patient Advocacy, Chelsea Carbokas. Although we're delighted to have Bennett and Chelsea joining us, we do need to make the disclaimer that due to Taysha having a product in active clinical trials, it won't be possible for them to tell us everything we'd like to know at this time. Indeed, there will be questions they're simply not able to answer. That said, we look forward to hearing what Bennett and Chelsea have to say about their program. Bennett and Chelsea, thank you so much for giving us your time today. Welcome to Reverse Rett Bitesize.
Thank you, Rachel. Can you all hear me okay? Wonderful. It's so nice to see your faces. Thank you so much for joining us today. My name is Chelsea Carbokas. I am what is called the head of patient advocacy at Taysha, and I'll talk a little bit about what my role is in a moment. But I've been with Taysha for just over 2 years, and in that entire time, I've been lucky enough to partner with Reverse Rett. So we're so grateful to have this invitation to speak with you all directly today. So, as Rachel said, before we kick things off, just as a biotech company, there are some things that we need to share with you before we get started. So I just want to take a moment to acknowledge these legal obligations that we have as we're speaking to you directly as the community. So first off, it's important that everyone here understands that any discussions we have about our investigational gene therapy today are just that. They're investigational in nature. Our product has not been approved by any regulatory agency, including the MHRA in the UK, and there's no guarantee that it will be in the future. So one of the themes that you'll hear throughout our discussion today is we're still very early. There's still a lot to learn as we progress through our clinical trials. We will also talk generally with you about what eligibility is and why that criteria exists in a clinical trial. And we just want to call out that it's important for families to understand that that criteria is in place for specific reasons, and it will likely exclude many families from participating in our trials. So we just want that to be well understood. Our goal today is simply to provide educational information to you all. We want to answer any questions you may have specifically about Taysha's program if we are able to, but it is important that we as a company do not influence any decisions that you make about a potential treatment or a clinical trial. And you'll hear us throughout the discussion today, probably referring you back to your physician to have those discussions. So for those of you who are not familiar with Taysha, our company is focused on developing a gene therapy for Rett syndrome. We are so grateful to be here, as I said today, to partner with you all as we progress this program. And we just want to take a moment to acknowledge the Rett community and advocacy groups such as Reverse Rett, who are the experts. And there's so much we can learn from you. So you truly are the foundation for every decision we make at the company. And we strive to incorporate your perspectives and your thoughts and your hopes into our work so that we can keep your best interests at heart. So over here on the slide, I just have a few areas that we focused on to date. We've done some research, which I'll get into in a moment, to learn about your lived experience and what are the most challenging symptoms that influence your day-to-day life. How can we then translate those into capturing what might be the most meaningful changes that you would want to see in a loved one if they were to benefit from a potential therapy? What are some of the things that families want to learn more about related to gene therapy and clinical trials? And I hope we're able to answer some of those in our presentation today. And then how can we reduce the burden on families who do participate in a clinical trial? So here is a list of just the primary patient advocacy groups that we partner with. So obviously Reverse Rett front and center here today, but they are really such a robust source of information for us and insight. They have such a tremendous pulse on the community, and the insights and perspectives they provide to us are truly so meaningful to us as we embark upon our journey. So I just listed here some of the ways that you can kind of see how we partner with the patient advocacy groups, helping to shape the protocols that we've developed for our clinical trials, learning how we can best support families as they're traveling if they're participating in the trial, integrating the perspective into the regulatory submissions. So, Rachel and others in the community have actually participated in our regulatory interactions in some way or form to really bring that community perspective to the forefront. And so many other ways. I couldn't list them all here, but I will mention also that, in the UK, it is important that when we're providing public updates about our program, that we go directly to the patient advocacy group. So a few years ago, maybe some of you may recall, Taysha fielded our first survey to learn about Rett syndrome. And we actually had 115 families in the UK who participated in our survey. So thank you to any of you who took the time to contribute. This survey was really essential in helping us to understand what are the most challenging symptoms that you experience with your loved one day to day. What would be the most meaningful improvements? How do we measure these? And how do we measure them in a way in our clinical trial that we're not burdening families who are participating? We also learned a lot about your overall perspectives of gene therapy, and what are some of the ways that you may want to learn more about gene therapy and clinical trials? So we took all these findings and we really have tried to share them back out in the community. So we are committed to developing posters like the one that you've seen on the screen here, bringing them to different conferences, sharing them with physicians. We're actually even working on a publication where we can publish some of these learnings just to share it out with the community. So again, thank you for taking the time to participate. There will likely be more opportunities like that in the future. So we appreciate anyone who is willing and able to share their experiences with us. So to the end of what we learned in here about families wanting to know more about gene therapy, clinical trials, and such, we are so grateful for Reverse Rett for hosting these webinar series. I think it's such a tremendous opportunity for us to share what we know about gene therapy and clinical trials with you. And again, we'll try to answer as many questions as we have as we are able to. So my final slide before I hand it over to Bennett for the meat of the presentation is just another reminder. We understand that time is extremely important for you and your loved ones, and we all work with that urgency in mind. However, we don't want to give false hope. Drug development does take a very long time, and we'll walk through kind of why it's such a stepwise approach and why things seem to take a little bit more time than we would all hope. So we will do our best to communicate the latest information as we are able. Our goal is always to be as transparent as possible, but we do have to apply those UK legal laws as we are embarking upon this. So again, thank you for understanding that we won't have all of your answers to your questions today, but we will do the best to answer as much as we can. So Bennett, I will hand it over to you.
Brilliant. Thank you. Thanks, Chelsea, and thanks everyone for giving us the opportunity to speak. I'm Bennett Maru. I'm the Chief Medical Officer with Taysha Gene Therapy. I'm an adult neurologist by training and background, and I'm based in the UK as well. So just going to move on into the overview of gene therapy. What is it, and what makes it such a nuance from traditional drug development as well? So typically, with gene therapy as it is today, it usually targets monogenic diseases. So diseases which would have, so diseases which usually have one gene that is the root cause of the disease as well. So at the moment, it's simpler to target a monogenic disorder than a disorder which has multiple genes involved as well. The aim of gene therapy, as we're working on it today, is to deliver a healthy copy, a healthy version of that gene, which will then enable normal protein production, which would help address the underlying cause of that disease. One of the ways that we're able to deliver that therapy, it needs to kind of be wrapped and enveloped into a delivery mechanism. So the cargo is the gene, and then it needs to be delivered, within whichever vehicle we choose. Now, at the moment, primarily most groups are utilizing viral vectors. Over 50 years ago, adeno-associated viral vectors were discovered. We know that they have a great affinity for certain parts of the body, and we use those as the delivery vehicle to give a gene in terms of a gene therapy as well. Now, the challenge with that, I mean, with it being a virus, is if you've never seen it before, your body is going to mount an immune response to it as well. So once you've mounted that immune response, it's fine for the first time. It makes it extremely challenging and difficult to redose again. And that's one of the reasons why there is a big concern around, redosing. If you've been given it once, could you give it again? And just purely for the fact that they are viruses. And I think that's something just to make sure that we wanted to get that across primarily. So what are the underlying causes of Rett syndrome? I understand that you probably all know a very great deal about this as well. But with Rett syndrome, it's caused because there's either a mutation, a deletion, an alteration within the MECP2 gene, which creates a protein MECP2. That protein is very important for neuronal maintenance in terms of how your neurons are working within the brain, how it's firing, and how it's able to conduct itself. Well, how you can be within normal human development. The goal of the gene therapy is to deliver a healthy copy of the MECP2, which would then help compensate for those MECP2 genes which are mutated within Rett syndrome. But also with Rett syndrome, as you know, you can have a condition which can lead to too many MECP2, so MECP2 overexpression, so there's MECP2 duplication, and then not enough is Rett syndrome. Okay. So some important facts about gene therapy. Kind of address this at the beginning as well. It is a one-time administration for a viral therapy, as it is at the moment as well. Patients that have taken part in this, in one gene therapy trial, are not likely unable to take part in another gene therapy trial as well, which uses a viral vector. There is a risk of immune reaction following gene therapy, and this is because the body may think of the virus as an intruder, as it would, and it would develop, and it would mount an immune response. We have to be mindful of that, and there is some immunosuppression that is given to the participant as well, just to help mitigate for that. The patients are monitored extremely closely, especially if it's a first-in-human gene therapy. We will monitor the blood as much as possible to monitor for immune reactions, for an allergic reaction. We will also monitor vital signs. I think the most important thing is safety is paramount for in any development process whatsoever. I mean, for us as well, we just want to make sure that this is extremely safe, and That's the primary aim of a gene therapy first in human clinical trial. Okay. So what is the aim of a phase 1/2 study? Now, in traditional development, which is not gene therapy, you have 4 phases of development: phase 1, 2, 3, and 4. Phase 1 is actually a healthy volunteer study. You're giving a drug, to a healthy volunteer just to make sure that there are no adverse events. We're not able to do that within gene therapy. And in gene therapy, you have to have the disease, for us to be able to even assess if it's safe. So that's why it's a phase 1/2 study. So you're assessing the safety as well as assessing for early efficacy, if possible, as well. So you can't give a gene therapy to a healthy volunteer that doesn't have the gene defect as well. So I think that's one of the biggest, most crucial defining factors in terms of normal development of normal drugs as well. And because of that, we do limit the number of participants that are able to take part in that study in the first instance. In the first instance as well, we want to ascertain which dose would be the most beneficial. So we're always leveraging doses which we believe will be beneficial. Can we go higher? Do we need to go lower? And again, we don't want to expose people to unnecessary risk, which is why there is always a small number of patients that are always within that study. Once we've completed that dose finding phase, we've understood that this is what the safety is. We then understand what some of the early efficacy as well. That's when you then move on to your later stages of development, like phase 3, and where it's opened up to a broader population. This is one of the reasons why we have eligibility criterion as well. So are you able to take part or not able to take part in a study? It's designed to make sure, again, that we're trying to minimize the risk as much as possible, trying to minimize any adverse events that could happen as well. So that's why you do see inclusion and exclusion criterion. Okay. So moving on to who would carry out the research as well. So firstly, it would be a sponsor. The sponsor is, in this instance, Taysha, responsible for defining the study, is responsible for ascertaining that they have all the correct approvals for the study, a CTA clearance from the MHRA, for example. We are then also responsible to ensure that the sites that are chosen are qualified, they have the right equipment, and they are able to ensure the correct monitoring of the patients as well. So that is the responsibility of the sponsor. The sponsor is the one that bears the ultimate risk and is responsible for all the risks as well. So the buck stops with the sponsor, for want of a better phrase. Once you have your CTA clearance, we then have to go through ethics. So ethics committees have to review and approve. Then the local site may have a separate ethics committee as well. So we have to ensure that we're working side by side, not only with a regulatory agency, but with a potential site as well, to ensure that the study is fit for purpose, and the site that is carrying out is able to carry out the study as deemed necessary. So the sponsor would identify the sites. We would then choose who we would want to work with. The principal investigator is usually the person that would be in charge of that study within that trial site. It would be us as the sponsor assessing these PIs, and then the PI would be the one that prepares the trial protocol as per the study, and they would then lead the entirety of a study team, and then they would report what their findings are back to the study sponsor, but it's always in a blinded manner. Just as for us, the safety of the patient is paramount. It's exactly the same for the PI as well. For them, the main purpose is to ensure the health and well-being of every study participant. The sponsor here is not allowed to interfere. The clinical decision made by the PI is what sticks and what has to be held true. We have no control over that, nor should we, because again, the patient safety is paramount. The participation within a study of a participant is ultimately determined by the principal investigator as well. And we as the sponsor would always remain blinded from any identifiable information or anything such as that as well. So participating in any study is does require a lot of commitment, and we appreciate that from the family and everything Chelsea's mentioned in terms of, you know, how we want to engage with the community, make sure we're doing right by the community as well. And we don't want to be the additional burden. Whilst we know that there is great potential here, that will, you know, have to be weighed up as well. So we are truly focused on, you know, patient-focused drug development. We need to hear what you guys have been telling us, just to make sure that we're not having an additional burden either. But in general, just because of it being a gene therapy and it being investigational, there is going to be some additional assessments that would need to occur. So in the initial period, there is going to be a requirement to be close to the site, or, you know, we would help support, you know, being closer to the site as well, because there are going to be many site visits. They're always to a preset schedule and as the protocol as well. So the first part would be screening. The screening is when the potential participant would undergo some assessments and tests. We would then determine full eligibility as per the inclusion and exclusion criterion. If for whatever reason it may be, they're ineligible, they would not be able to partake in the investigational gene therapy study. If they are, then we would move them forward into the baseline period. And then with the baseline period, every participant that is eligible would undergo the initial tests and assessment before they would actually receive the gene therapy. In some instances, there is randomization, in some there isn't. If there is randomization, the participant will either be randomly assigned to receive an investigational therapy, or they would be assigned to a delay to treat cohort. So the two groups would be called cohorts. And if you are randomized to delay, you would receive the gene therapy upon whenever that time frame period is for the delay. There is ongoing observation. There's ongoing observation for a year or two with part of the main study. Then there's a long-term follow-up. And due to it being a gene therapy, a lot of regulators would like data to be collected over a longer period of time. And within Europe, it's still 15 years as well. So 15 years worth of data still needs to be collected. So there would be an ongoing commitment to take part in an investigational gene therapy program. Okay. So how are we determining if the therapy is safe and if it's working? So as I mentioned before, it's important to remember a phase 1 therapy is the first time the product has been put into humans. That piece is key. It is structured primarily as a safety study. It always has to be, again, because it's the first time it's being put into humans. And, you know, safety is our primary concern in all of this. From an initial part from a phase 1/2 study, once a patient has been dosed, there is always a period of waiting before the next patient is allowed to be dosed as well. It allows us to understand if there are any safety signals that we need to be monitoring for, if there are anything that, you know, we hadn't anticipated. So there is always a time delay between dosing one patient to another patient. In that interim period, we are collecting safety information. The site is collecting safety information. It's put into the system. It is reviewed by the PI. There is a medical monitor which would be reviewing the data as well. It's all then captured and put together, and then it is presented to an independent data monitoring committee. So it's a safety board. They are not associated to the study. They are not associated with the sponsor either. They truly are an independent committee who are pulled together to review the data to understand the safety profile of the product. The IDMC, the Independent Data Monitoring Committee, they are experts in their own right. They have different expertise in different areas, and we'd like to pull them together. And they would review the data. They would look at the safety data initially. That is their primary aim. They would look at some of the potential efficacy as well, and they look at the conduct of the study as the study is ongoing. Once they've reviewed the data, they then make the decision and recommendation whether another patient should be dosed, or if we, in a case if there's, you know, multiple doses, if you've ended that one cohort, if you then want to move to a second dose level as well. All of these safety parameters are put into place, and there is an interval of about 6 weeks typically from, you know, one participant to another participant being able to be dosed as well, because that's the minimum amount of data that would be required. As you can imagine, with all of the assessments that are being conducted, even though it's a relatively small number of participants, there is a lot of data that is captured from tests and assessments. Some of them are included, for Rett syndrome, would be specific to Rett syndrome as well, looking at movement communication. There'd be ECGs, there'd be echoes, to monitor for the heart. You'd want to make sure things in the brain are okay, so MRIs would be conducted. We would be conducting motor and sensory examinations. We would be looking just to make sure that there are no adverse events, that you wouldn't expect elsewhere as well. And then we'd be looking at communication abilities and such as well. Some of the assessments that we do require as part of the safety monitoring do require sedation or some general anesthesia as well. And all of this is just to make sure that we're doing things appropriately as well. And all of this would be put into an informed consent form. So everything, and a patient information leaflet as well. So everything would be fully transparent. You'd be discussing it with the trial site, with the PI as well. Okay. So what's the regulatory pathway? The regulators are involved from the beginning. They're there to determine whether you can actually run a human study or not. They would make sure that they would monitor what the information is as you move through the phases of the trials, and then eventually they would be there to determine whether the product is actually safe and it is efficacious, and if it's available, for wider population use. So they actually give the approval for the drug to be used. So that they're the group of people that we would need to work with the closest. So the regulators, whether it's the FDA in the States, EMA for Europe, and MHRA, they review the preclinical data as well as the clinical data. At certain points during the phases of development, the sponsor would provide results of the trials to the regulators. They have their medical and scientific experts who review the information. And then this is where they would then decide whether they approve a treatment for wider use, or if they need more information, they could ask you to do another study as well. Their job is, again, to primarily look at the safety of the program, to see if the benefits outweigh the risks, but they also do want to see if there's an improvement in patient care as well. But prior to any of the regulatory approval, all potential therapies can only be conducted in the setting of a clinical trial. So just to go through quickly the REVEAL studies. Now, we are currently conducting investigational studies in Canada and in the US. Again, important to note, they're first in human studies, and we are not able to make any definitive statements or conclusions around interim findings of the studies to date. So these studies are designed to evaluate whether this gene therapy is safe, whether it's tolerated, and is it actually beneficial as well. These studies are designed to give us the information we need to be able to test to a wider population in the future as well. So the two studies currently, so the one in Canada, which is the adolescent and adult study, and then the one in the US is our REVEAL pediatric study for children 5 to 8 years. And as you are aware, the UK granted approval for us to run the pediatric REVEAL study in the UK. We are not currently enrolling as of yet. So what is the timeline for enrollment in the UK? So there are several things that we need to step through. So now that we have MHRA approval, we now then have to work closely in determining, working with the potential sites, working with the potential PIs. There is a lot of administration that would need to go through as well. So we actually need to make sure that there are some local ethics committees that would need to review. We need to make sure everything is in place with the potential sites as well in terms of contracts. All of that will take time. There will be some training that would be required because there are some things that we would be utilizing and we want things collected in a certain manner, so we need to be able to train the sites as well. All of this can happen, all of this needs to happen before we're able to actually say that a site has been accepted and before it's been initiated to enroll a patient as well. So once the clinical trial has been initiated, we would obviously notify Reverse Rett and other advocacy groups in terms of where we are and what stage there are. And if for information with regard to sites, we would ask you to look at the clinicaltrials.gov website. But if there are any questions, anything with regard to eligibility, it would be great, for the families to discuss with the principal investigator. And that kind of leads us nicely just to say thank you. Thank you for allowing us to speak, and just thank you for giving us the feedback that you have given us as well. It is tough, this is challenging, and I know people are kind of sometimes chomping at the bit as well. So we thank you for your patience as well.
Much appreciated, and thank you to you and Bennett, Chelsea. That was just all so interesting, and it's great to see it from a completely different perspective. Even some of the information that I think we may have shared before, it's just helpful to see that from a different perspective, from the industry perspective in particular. And also just I'm just really grateful to you both for being here. And I have actually written down a few questions, although I'm supposed to be the person that's making sure you don't really get off the cuff questions. I feel a bit guilty, but I think it's just listening to you, Bennett, has really stimulated a lot of questions in my mind. Catherine, why don't you go ahead with the questions that were pre-arranged, and then we'll see if some of mine get answered. And in the meantime, if you do have questions and you'd like to put them in the chat, everyone's been very quiet. But if you do have questions, you'd like to put them in the chat, one of Taysha's team will take a look at those and assess whether or not we'll be able to see if we can get an answer for you. Thank you.
So the first question, and I think this is probably furthermost in most of our minds, and you have answered it, but I just want to acknowledge Diane King, grandmother to Layla, asked if having been one of the very brave first participants to have the lower dose, would they be able to receive a top up? And I think that's been quite clearly answered, that due to the nature of how the dose is given, that wouldn't be possible. And then Kate McMaster has asked, she'd like to understand the reasoning in the proposed age groups so far for the adult pediatric trial in Canada and the US, and why are children aged 8 to 12 not being considered in the current cohort?
So as I mentioned earlier as well, from a safety and regulatory perspective for the initial phase 1/2 part of the study, we do want to be as narrow as we can be, and our initial study was 18 plus in Canada. For those reasons, until we've kind of worked through those age ranges at the moment, that's kind of where typically gene therapy studies would be as well, in the sense where having these narrow groups. That's not to say that in the future development, as we progress through the phases of the investigational product, Taysha wants to treat as broad as possible, and I think that's something that we would get to in the future. But for now, it's primarily safety driven as well. So want to stay within a certain band.
I think this is a good time for me to ask one of my other questions then. So if you do end up, because presumably, with these first groups of patients that are very small, presumably you'll have to open up to another clinical trial after this one. And if you do, after these two, if you do open them up, how many patients would you expect to expand to worldwide? What's the sort of normal amount that people would go to after doing this first kind of study? Can you answer that?
Yeah, and again, generalities as well. It generally all does depend, and I'm not trying to say that to fob anybody off. It all depends on the data that you've captured in terms of the effects that you're seeing and and then obviously there's statistics that would go behind that. We'd obviously need to have a discussion with the regulators as well, and this would be for any gene therapy product or any drug development in general. So it all has to be a data-driven decision. At the moment, it's too early for us to say what those could be. They could be small, they could be large. I don't think anyone could tell you, but the data we'd collect, from the 2 phase 1/2 studies at the moment would inform the decision for, the next phase of trials ultimately.
Great, thank you. In other disease groups, have you seen any evidence that it's possible to go to the regulator even after a sort of smaller gene therapy trial? I mean, I think that the problem is because we're in such uncharted territory with these gene therapies that are emerging. We've only got a handful here in the UK. I think nobody really knows what to expect, which is why it's so helpful to be able to have these kinds of conversations. That's a question that's kind of turned into a comment. I think I'll leave it there. There's a few popping into the chat now, though, Catherine.
Yes, I'm just
Okay.
I'm just monitoring those. Chelsea, I don't know if you'd rather read through them and look at which ones
Yeah,
you're able to
sure,
answer.
Catherine. Thank you. So Bennett, I'll tee this one up to you. Can we talk a little bit more, I think, generally about the immune suppression? How long would it last? I don't know that we can go into details around the length of time, but would kids have to stay home from school? What are some of the considerations that a family should know about the immune suppression that is entailed in a gene therapy trial?
Sure. So yeah, obviously can't go into details in terms of the length and the doses and in that sense, but in generalities, this would be a very important conversation to have with the PI at the site as well. They will know the participant, just as well as the family would. During now coughs and cold season, it would be mindful, just to make sure that take that into consideration as well. It shouldn't necessarily stop going to school, but again, it depends on how the participant is at the time as well. It's discussion genuinely to have with the PI as well. We do have some information that would be developed, or that is developed and would be given to trial participants around immunosuppression as well. We don't want anyone getting sick ultimately.
Yeah, Bennett, you bring up a good point that I want to make sure is clear here. So the REVEAL pediatric study is the same study in the US and the UK. So when we talk about this first part of up to or approximately 6 participants, and we've already dosed 1 in the US, that leaves 5 spots left across the US and the UK. So that kind of gives you an idea of how small of a number of participants will be in this first part of the study. So just want to make sure that that's clear. Next question was, how will it work regarding age and eligibility, where a patient may be eligible at the start, but several months later, by the time the trial starts, they could be outside the age range?
Unfortunately, from a consenting perspective, it would all matter with regard to what the eligibility criterion is at the age of consent. So in that scenario, they would be ineligible.
Next question is, with the variation of Rett syndrome on people in relation to what a young person can and can't do and is currently experiencing, is this a factor with trial selection? So I think we're getting at, the level of severity, and is that part of our inclusion or exclusion criteria? And hopefully I'm not putting words into this person's mouth.
So there are clear criterion in terms of for inclusion exclusion, again for any gene therapy as well, in terms of especially for like monogenic diseases, there's clear criterion that should be there around what the genetic mutation is, what it impacts, and also what the severity at enrollment is as well. So as mentioned before, we want to try and keep minimize the risk and assess what the positive effects are as well. So for a phase 1/2 study, it does have to be fairly narrow in terms of what's eligible in terms of severity and genetic status. Again, that doesn't mean that again, for the phase 1/2 piece, it doesn't mean that when you get to the later stages of development that it's not going to get broader. But for now, it has to be quite narrow.
And I just want to do a reminder that eligibility criteria is established in our protocol, but that protocol is managed by the clinical trial site. So once the clinical trial site is announced, families, if they're interested in knowing whether they may be eligible or not, they should reach out directly to that trial site and the principal investigator. I think that's all the questions in here. Bennett just sent me a quick reminder. Yes. So I will make sure, Rachel, that you have the clinicaltrials.gov link. I think you've probably already shared it out, but there's more detail on that page if families want to look at some of the inclusion exclusion criteria.
Okay, so thank you for that. Yeah, I can't see any other questions either. We will put all the links on the information that we send you when we send you the recording, once we've had a chance to top and tail it and take all of my bloopers out. So do bear with us. It might take a little while to get that out to you. As Chelsea has already said, big up the Rett registry. If you don't already have your person with Rett syndrome or your adult or child registered, literally just email me. It's catherine@reverserett.org.uk and I can send you a link. We've done a lot of work over the summer getting it up to the 21st century. It's now really easy to register and log in. And we do ask that you put pictures of your person with Rett syndrome on. It's lovely for us when we're looking at data if we can see pictures. The other things that I need to tell you about is a very, very exciting day coming in June. The 1st of June, we're holding a LEAP Education Day. So LEAP is Learn, Educate, Advocate. And what was the last one, Rachel?
It's actually empower.
See, I don't prepare. That's really obvious, isn't it? So we have emailed out over the last few days. It's gone on social media. We've emailed out to everybody who's already subscribing to our newsletter, and we've emailed today to everybody on the registry. Numbers are limited, and we've today adjusted the amount of tickets we have available for people with Rett syndrome to attend the event, because it is a family day. But we are limited on numbers, and the numbers are going very quickly. So if you are interested, it's a completely free event. Have a look in your junk email if you don't think you've received an email from us today, because everything's gone out now. And again, if you can't find it and you want to come, email me and I'll send you the link. It's very, very easy. The next bite-sized session, these feel like they come around very quickly, is the 15th of February. And we're waiting to confirm who the speaker is, but in true Rachel fashion, it will be someone as amazing as today. So keep an eye out for those emails as well. We will be sending you details as you've attended today's event to register for the next event as well. So from me, that's everything. Thank you all for coming today. You know, these events wouldn't be possible without your participation. And I know as parents, we're all going, come on, come on.
Hurry up.
But thank you for your patience.
And I think we should also just say a big thank you as well to Chelsea and Bennett and everyone from Taysha for being part of today's session and for all of the work that you're doing on behalf of everyone with Rett syndrome worldwide. We appreciate what you're doing so much. We understand it all takes time and the care that you have to take, but we really, truly appreciate how patient-focused you are at Taysha and the partnership that we have with you, and that the input that we've been able to provide as you've developed the program. So thank you all very much.
Thank you. Thank you so much.
Thank you, Reverse Rett.
Thank you, everybody.
Thank
Thanks.
you.
Bye. Bye.
Thank you.